Bismillah ar-Rahman ar-Rahim. Today we will talk about the chronic inflammation. Now, in discussing the chronic inflammation, we will discuss the causes of chronic inflammation and we will discuss the inflammatory cells that are involved in chronic inflammation. Chronic inflammation, as compared to acute inflammation, it is comparatively, it starts slowly and take days, and it may remain for months and years. And chronic inflammation always results when the infection is very severe and when it is very prolonged, particularly when the eliciting agent resists eradication. For example, Mycobacterium tuberculosis, which causes tuberculosis infection, and Treponema pallidum, which causes the disease sepsis, and certain virus infections, for example, hepatitis B and hepatitis C viruses. And chronic inflammation also results when the inflammation is directed inappropriately. For example, in autoimmune diseases, what happens that our body or our immune system produce antibodies against the own body tissues, and they act as uh, they behave the body tissues as the non-self. In other conditions in which there is an exaggerated immune response, for example, the bronchial asthma and other types of allergies in which the uh, apparently normal environmental antigens and allergens cause a person who is genetically susceptible uh, uh, induce the environmental allergens and antigens induce disease. So in these two situations, the autoimmune diseases and allergy or hypersensitivity, we say that the immune system is inappropriately directed. Now, in if we compare chronic inflammation to the acute inflammation, usually the acute inflammation mostly it may play a beneficial role because it is short-lived and it is self-limited, and it is uh, comparatively it causes less tissue damage as compared to chronic inflammation. And the outcomes of acute inflammation, mostly it is in the form of resolution in which the uh, anatomical and pathological integrity of the tissue is restored, and uh, at the end we get a normal anatomical and pathologically functioning tissue. In chronic inflammation, there is damage, excessive damage to the normal tissue. Now, chronic inflammation results when the inflammation is prolonged, or if there is some problem which causes poorly controlled inflammation. And the difference between the acute and chronic inflammation is that the acute inflammation is fast, it occurs in minutes to hours, while the chronic inflammation is comparatively slow and it takes months and years. And the inflammatory cells involved in acute inflammation are mainly the neutrophils, while in chronic inflammation the inflammatory cells are the mononuclear cells, uh, monocytes, macrophages, and lymphocytes, and other inflammatory cells. The tissue injury and fibrosis, it is usually mild and self-limited in acute inflammation. And in chronic inflammation, the tissue injury is often severe and progressive, and it causes a lot of problems. Now, when we come to the local and systemic signs in acute inflammation, these are very prominent. There is high fever, temperature, pain, swelling, and uh, swelling that occurs due to exudation and edema formation. But in chronic inflammation, the signs and symptoms may be initially subtle and less prominent. Now, when we say this is chronic inflammation actually it has got three components. And when we find these three components, we say this is chronic inflammation. As I said, that chronic inflammation is of prolonged duration and it takes weeks to years. And the inflammatory cells in chronic inflammation, usually these are mononuclear cells that include macrophages, lymphocytes, and plasma cells. Whenever a tissue is injured by an external trigger, initially there is infiltration of the neutrophils, polymarbonuclear leukocytes, but after 24 to 48 hours, these neutrophils are replaced by the mononuclear cells, which are macrophages, lymphocytes, and plasma cells. Now, in chronic inflammation, there will be tissue injury and there will be tissue destruction. And this tissue injury and tissue destruction is induced by the inflammatory cells, which secrete inflammatory mediators and form reactive species and secrete lysosomal enzymes which degrade and damage the tissue. The third and most important component, and which is a major difference between acute and chronic inflammation, is that in acute inflammation, mostly there is resolution occurs. The tissue healing and repair, which is characterized by the scar tissue formation, which may give a little bit disfiguration to the tissue, is the component of the chronic inflammation. In chronic inflammation, what happens that there is fibrosis, where the damaged tissue is replaced by fibrous tissue. There is proliferation of the fibroblast that synthesize collagen, and also there is new blood vessel formation in chronic inflammation that is known as the angiogenesis. Now we come to the causes of chronic inflammation. Chronic inflammation, in most situations, it is a continuation of the acute inflammatory response, where the acute inflammation is unable to resolve the problem, right? So what are the conditions in which acute inflammation will be unable to solve the problem, particularly when the injurious agents, they are persistent? For example, mycobacterium tuberculosis, Treponema pallidum, and certain viruses, they are difficult to be eradicated by the acute inflammation, and ultimately the acute inflammation turns into chronic inflammation. The second situation in which inflammation will uh, go into its chronic form is when there is some interference to the normal process of healing. So, uh, keeping these situations in mind, we say that the causes of the chronic inflammation may be those microorganisms that are difficult to be eradicated and persistent infection and inflammation. For example, as we have talked that uh, uh, when the inflammation is inappropriately directed, in the form of autoimmunity and in the form of exaggerated reactions called the allergy or hypersensitivity. So in that type, usually type 4 or delayed hypersensitivity reaction occurs. For example, if the person is infected by the mycobacterium tuberculosis, and if the person suffers from the disease syphilis, and if a person gets certain type of viruses and fungi, what happens that the cellular response occurs in the form of uh, delayed hypersensitivity. As we know that there are four types of hypersensitivity reactions, hypersensitivity reaction 1, 2, 3, and 4. The fourth hypersensitivity reaction is also known as the cell mediated hypersensitivity, because in this hypersensitivity reaction mainly the T lymphocytes are involved. In the case of exaggerated immune response to normal environmental allergens or pathogens, that may occur only in those people who are genetically susceptible. For example, the bronchial asthma. In autoimmune response, in which the immune response is inappropriately directed, what happens that the body immune system reacts in the form of antibody or in the form of T cell mediated reactions, and it will cause, uh, it will mount an inflammatory response to the body's own tissues, and they consider them as the non-self. The examples of diseases like rheumatoid arthritis, irritable bowel syndrome, for example, uh, uh, ulcerative colitis, Crohn's disease, and the chronic autoimmune skin infection known as the psoriasis. The fourth situation which leads to the chronic inflammation is that when some non-degradable particles enter into the body, and most of the time these non-degradable particles are inhaled. Examples are the disease silicosis and pneumoconiosis. In silicosis, the silica particles, they are inhaled in those, particularly those people who work in such like industries and situations where there are silica particles. So the cell, human cell have no machinery to degrade the silica particles. The similar situation occurs in coal worker pneumoconiosis, uh, also known as anthracosis, in which the carbon particles which are inhaled, and again the body cells have no machinery, the phagocytes have no machinery to degrade carbon particles, and initially they are taken up by the macrophages and they remain there for long times and uh, induce repeatedly the inflammatory responses, and the result is the disease like silicosis, pneumoconiosis, including anthracosis. Now, recently it has been observed that the mild form of inflammation uh, occurs in the body, and this is the major cause of chronic inflammation behind diseases which are apparently not known to be inflammatory in nature. For example, Alzheimer's disease, in which uh, associated with age, there is mental retardation, type 2 diabetes, which is characterized by hyperglycemia, and the atherosclerosis. Particularly in atherosclerosis, what happens that there is initially an injury to the vessel which results in the atherosclerotic plaque formation, and that are responsible for severe diseases like myocardial infarction and hemorrhagic strokes or cerebral strokes. Now, coming to the cells of the chronic inflammation, we have macrophages, which are the dominant cells in chronic inflammation. We have lymphocytes, which are B lymphocytes and T lymphocytes, and the plasma cells, which are derived from the B lymphocytes and which are associated with the secretion of antibodies. The eosinophils, which is one of the three component of three granulocytes: neutrophils, basophils, and eosinophils. The eosinophils are characterized by the spectacle shaped nucleus, and eosinophils are involved in parasitic infections and allergies. The mast cells, which are located around the blood vessels, they are the major source of histamine, and we know that histamine is strong vasodilator and it increases the vascular permeability. The neutrophils, which are particularly cells of the acute inflammation, and in some situations the neutrophils are seen in certain chronic inflammatory conditions. Now, first we talk of the macrophages. The macrophages are part of the mononuclear phagocyte system. Initially, it was called as reticuloendothelial system. Now, the monocytes, the macrophages, actually they are the tissue cells derived from the circulating blood monocytes. The monocytes which are produced in the bone marrow, they circulate in the blood, and here we say that they are here in an inactive form. But when tissue injury occurs, when there is an inflammatory response, they leave the blood vessels uh, by the way of relatively similar to the way in which the neutrophils leave the circulation through diapedesis and go into the outside of the blood vessel into the extravascular tissue. Now, in the extravascular tissue, the monocytes, they become macrophages, and macrophages are more powerful cells that fight against infection and immune reactions. And the same macrophages, when they are present in the liver, we call them as the Kupffer cells. In the spleen and lymph nodes, we call the macrophages as sinus histiocytes. If present in the central nervous system, the macrophages are called the microglial cells. And in the lung, we call the macrophages as the alveolar macrophages. So this is the macrophage that has got different names according to the different tissues where they persist. Now, the macrophage to be fully active, it must first be activated. And for the activation of macrophages, there are two pathways. So, macrophages must be activated before they take part in inflammation and immune reactions. There are two pathways by which the macrophages can be activated. One is known as the classical pathway, and the other is known as the alternative pathway. In classical pathway, the macrophage, when in uh, it is activated by the microbial products like endotoxin and uh, polysaccharides and other components of the microorganisms, and they can also be activated through its classical pathway by the T lymphocytes, and signals from the T lymphocytes in the form of particularly interferon gamma, it will activate the macrophages uh, to perform its role in inflammation. Now, certain foreign substances in the form of crystals and particulate matters, they can also activate the macrophages. Now, here we have to notice two important points. When the macrophages are activated by the classical pathway called M1, so they will induce inflammation. They have got their major role in inflammation because these classically activated macrophages, they will release lysosomal enzymes to kill and degrade microorganisms. They will react, they will form uh, in the through the way of oxidative burst in the pagolysosome, for example, nitrous oxide, and reactive oxygen species. And we know that these are free radicals and they will cause uh, destruction of the microorganisms. Now, they by secreting certain cytokines also, the macrophages stimulate inflammation and amplify the reaction, and through lysosomal enzymes and reactive species in the form of nitric oxide and reactive oxygen species, they kill the ingested organisms. And we know that the reactive oxygen species and nitric oxide, they will cause peroxidation of the uh, layers, lipid layers of the microorganisms, and they will also cause denaturation of the proteins, and in this way they will kill the ingested organisms. So we come to the conclusion that when macrophages are activated through its classical pathway, they will have their major role in inflammation and killing of the microorganisms. On the other hand, macrophages can be alternatively activated, and this alternatively activated macrophages are known as the M2. They are caused by the interleukin 4 and interleukin 13. And interleukin 4 and interleukin 13, they are secreted by the T lymphocytes and the mast cells and the uh, eosinophils. So they will activate the macrophages known as the M2 through an alternative pathway. Now, macrophages that are activated by the alternative pathway, they will play their role in tissue repair and fibrosis, because macrophages activated through this pathway will secrete uh, tissue growth, transforming growth factor beta, and we know that transforming growth factor beta take part in the tissue repair and proliferation of the fibroblasts and fibrosis. Now, here in this slide, we make the two pathways together. Here we can see that the macrophages, if they are activated by the microorganisms or by the interferon gamma, it will take part in the inflammation. And these macrophages, they will secrete, they will form reactive oxygen species, nitric oxide, and lysosomal enzymes. And these reactive oxygen species, nitric oxide, lysosomal enzymes, they are used in killing of the microorganisms. So they take part in microbicidal actions, phagocytosis, and killing of the bacteria and fungi. Additionally, these macrophages secrete interleukin 1, interleukin 12, and interleukin 23, which are chemokines, and they take part in inflammation. So here we come to the conclusion that classically activated macrophages, they have their role in inflammation and killing of the microorganisms. They kill microorganisms, for example, bacteria and fungi, and they amplify the role of inflammation by secreting various inflammatory mediators and chemokines. On the other hand, we can see on the right side that those macrophages that are activated by the alternative pathway and known as the M2, they secrete two types, major types of uh, mediators. These macrophages, if they secrete transforming growth factor beta, for example, it will induce tissue repair and fibrosis. It will induce proliferation of the fibroblasts and take part in healing and repair. And along with the transforming growth factor beta, if the macrophages also secrete into interleukin 10, so in this situation it will have an anti-inflammatory effect. And we come to the conclusion that the alternatively activated macrophages, they take part and they have their role in tissue repair and fibrosis. Now, here we come to the summary of the functions of the macrophages. They do phagocytosis. By that we mean that they engulf or ingest the microorganisms, forming the phagosome, and the lysosomes with hydrolytic enzymes is present in the lysosomes of these cells. The lysosome, it unites with the phagosome to form the phagolysosome, and the phagolysosome is the site where these cells kill the microorganisms through various enzymes and reactive species. The second function, which is the tissue repair, and this particularly occurs when the macrophages are activated through the alternative pathway, and it will lead to the proliferation of the fibroblast and collagen synthesis, and finally the scar formation and fibrosis. The third function of the macrophages is the secretion of cytokines. For example, it secretes tumor necrotic factor interleukin 1, which directly stimulate and activate the neutrophils, and various chemokines which are involved in the process of chemotaxis by pulling the inflammatory cells, are inducing a directional movement of the uh, microorganisms and the inflammatory cells towards each other. And they also secrete the eicosanoids, which are uh, arachidonic acid metabolites and prostaglandins and uh, lipotrienes. So we can say that the macrophages are the cells that are central to the initiation and propagation of all inflammatory reactions. The same macrophages, when activated by the alternative pathway, so the transforming growth factor along with interleukin 10, it acts as an anti-inflammatory agents, and they work against the inflammation as an anti-inflammatory. Now, one important function of the macrophage is that whenever they face the microorganisms or dead tissue, what happens that they ingest the microorganism, for example, a bacterium or a dead tissue, and within the macrophage, this microorganism is broken down into pieces, and with a piece, these macrophages present these microorganisms, bacteria, fungi, or dead tissue to the T lymphocytes. And here we say that now they are the antigen presenting cells. The antigen presenting cells mainly are the macrophages, dendritic cells, B cells, and they present the antigen, part of the processed antigen, to the T lymphocytes. and this is a red flag for the T lymphocytes. And through this type of presentation by the antigen presenting cells, they activate the T lymphocytes and B cells, and thus setting up a feedback loop that is essential for defense against many microbes by cell mediated immune response. So there are two types of immune responses. One is the humoral or antibody mediated cell response, in which the antibodies are secreted mainly by the lymphocytes, which are derived from the secreted plasma cells, which are derived from the B lymphocytes, and the other is the cell mediated response, in which the T lymphocytes take part. Now, coming to the lymphocytes, there are two major types of lymphocytes. One is known as the B lymphocytes, which are involved in particularly in the acquired immunity by secreting uh, uh, antibodies. So what happens that the B lymphocytes, they migrate to the site of infection and tissue injury, and as a part of the acquired immune system, these B lymphocytes, they develop into plasma cells, which are oval-shaped cells with eccentric cartwheel nuclei, and these cells, they secrete antibodies. Right? On the other hand, the T lymphocytes, when the antigen-presenting cells like a macrophage present a part of the antigen to the T lymphocytes, they get interacted, and the macrophage secretes certain cytokines which activate the naive T cells. And the naive T cells, when they should be activated before they perform their role in inflammation and in the immune mediated processes. Once the naive T cells are activated, they will get transformed into various types of T cells. For example, we know that we divide T cells, particularly the small lymphocytes, into T helper cells, effector cells, T suppressor cells. And here in this situation, when the naive T cells, when it interacts with the macrophage or the antigen presenting cell, which secretes cytokines, which cause differentiation of the naive T cells into Th1 T cells, Th2 T cells, and Th17. All of these are helper T cells. Now, then next comes the role of these differentiated T lymphocytes. The Th1 cells, they secrete interferon gamma, which is a cytokine, and through the secretion of interferon gamma, the Th1 cells activate more and more macrophages. And this happens in the classical pathway of macrophage activation. Now, the Th2 cells, they will secrete interleukin 4, interleukin 5, and interleukin 13. Also, these cytokines, they will activate macrophages, but here they activate macrophages in the alternative pathway, and the macrophages that are activated by the alternative pathway, they take part in tissue healing and fibrosis. Additionally, the Th2 cells also secrete the cytokines, as I said, the interleukin 4, 5, and interleukin 13, and they also activate the eosinophils, which are part of the granulocytes, and these are the cells that are involved in hypersensitivity reactions and additionally in parasitic infections. The Th17 lymphocytes, they will secrete Th17 and other cytokines, and they will uh, recruit neutrophils and monocytes into the reaction. They will promote the recruiting and activation of the neutrophils and monocytes. Now, here we come to the conclusion that the T lymphocytes and macrophages, they interact with each other to take part in inflammation. They amplify each other. So in here on the right side, we can see that the uh, macrophage is when activated, it will present part of the ingested microbe or dead tissue to the T cells. And here the cytokines, for example, interleukin 12, 6, and 23, they are secreted by the macrophages. And in this way, the macrophage is used to activate T lymphocytes. Now, the T lymphocytes, once it is activated by the macrophage, it will get transformed into helper T cells 1 and, for example, helper T cells 2, Th1, Th2 and Th17 cells. Now what happens that these activated T lymphocytes, they will secrete interleukin 17 and tumor necrotic factor, and in this way they will cause recruitment of the neutrophils, and on the left side we can see that the macrophage directly, this is an indirect way of leukocyte recruitment uh, and to take part in inflammation. In the direct way, the macrophage also secrete tumor necrotic factor and interleukin 1. And here in this situation, on the right side, the macrophage is directly involved in the leukocyte recruitment uh, and amplifying the inflammatory reaction. Now, the macrophages, activated macrophages, they also secrete interferon gamma, and the interferon gamma activate further macrophages, and in this way the reaction is amplified, and from this slide we can uh, conclude that the macrophages and the T lymphocytes, they interact with each other to amplify the inflammatory reaction. Now the other cells that take part in inflammation are the eosinophils and mast cells. The eosinophils actually, the three types of granulocytes, the neutrophils, basophils, and eosinophils. So these are granulocytes. They have granules in their cytoplasm, and their nucleus appears as spectacle-shaped nucleus. And the eosinophils take part in parasitic infections around the parasites and to kill them, and they also take part in immune reactions which are mediated by the immunoglobulin E associated with allergies. The immunoglobulin E is the major antibody that take part in type 1 hypersensitivity reaction, in which the person once uh, infected by an bacterium are activated by an antigen, and which gives to the formation of antibodies, immunoglobulin E, which are attached to the mainly to the mast cells. And for example, if the same person gets reinfected by the same allergen or antigen, what happens that the antigen will bind to immunoglobulin E on the surface of the mast cells and eosinophils, and this antigen antibody reaction will result in the mast cells degranulation, and what happens that there is the release of histamine. So immunoglobulin E coated mast cells release histamine and other mediators to play a role in inflammation, because we know that histamine is a strong vasodilator, and uh, also it causes increased caliber of the blood vessels and increase permeability, and uh, in severe situations, if the type 1 hypersensitivity reaction, which is also known as the anaphylactic reaction, if it is very severe, what happens that so much histamine is released into the circulation that it causes so much vascular dilatation that the person may go into hypotension and hypotensive shock, and in severe situations, if not treated properly, a person can die of this severe anaphylactic reaction, which is basically induced by the immunoglobulin E attached to the mast cells and its interaction with the antigen, which causes degranulation of the mast cells with the release of large amounts of histamine, which cause vasodilation, dilates the blood vessels, and can lead to severe hypotension. Thank you very much.